Hello and welcome to Badger Talks Live, which brings exciting happenings, resources, and talent from your UW flagship campus to the people of Wisconsin and beyond. I am Quincy Rosemary, a cancer biology PhD candidate in my last year of graduate school, studying a human tumor virus in the McArdle Laboratory for Cancer Research in the School of Medicine and Public Health. I am originally from Indonesia. My current research focuses on how Epstein-Barr virus, or EBV, takes over and manipulates a cell's nucleus to promote its own infectious cycle. EBV features some more in today's talk. During the month of September, Badger Talks Live is proud to showcase the latest cancer research in McArdle. I'm pleased to introduce Bill Sugden, who is the American Cancer Society Research Professor and the James A. Miller Professor of Cancer Research. Today, he will be discussing some of our research showing not only that viruses do cause some cancers in people, but in one case, two viruses act together to cause one lymphoma in some of us. While Bill and I have worked together for the last 6 years, he has worked on EBV for the last 50 years to understand what it contributes to human cancers. About 30 graduate students who have worked with him earning their PhDs now carry on their own research in this country and throughout the world. Bill will be answering questions at the end of the presentation, but feel free to post them in the chat at any point. Now please welcome Bill.
Thank you, Quincy. What I'd like to do is to describe to you a 20-year odyssey in which we've learned how two viruses cause one cancer. And those viruses are Epstein-Barr virus, Quincy introduced you to it, and Kaposi sarcoma herpes virus. And the cancer is primary effusion lymphoma. The work I'm going to describe is really a 20-year odyssey, and it's been done by multiple people, more than 7, but I'll describe the work of 7 of these people. And to put that in perspective, Odysseus made it home from the Trojan War to Ithaca in only 10 years. So this odyssey has taken more than twice his trip, and it's still ongoing. What I'm going to do is briefly introduce cancer to you, introduce viruses to you, spend a little bit more time introducing Epstein-Barr virus and Kaposi sarcoma herpes virus to you, and then spend quite a lot of time describing to you the work we've done that establishes that each of these viruses independently can cause cancers, and in one case, they work together to cause a cancer. So, cancer results from cells growing inappropriately and often colonizing inappropriate sites. And all cancers arise from multiple causes, that you've got to know. But primary effusion lymphomas, the ones I'm going to talk about, are unique in that they are caused by 2 viruses. So what are viruses? Well, they're parasites and they're absolutely dependent on cellular hosts. Viruses contain RNA or DNA as their genetic material. That RNA or DNA is usually protected in a protein shell. And this is the kicker. Viruses must infect cells to replicate, and they encode information to take over cellular functions to allow them to replicate. So the viruses I'm going to focus on are Epstein-Barr virus, I'm going to call it EBV throughout the talk, and Kaposi sarcoma herpes virus, I'm going to call it KSHV throughout the talk because it's a mouthful. So these 2 viruses cause about 300,000 new cases of cancer worldwide. And those cancers include lymphomas, carcinomas, and sarcomas. And so those are the 3 major types of human cancers. 300,000 is a big number, but it's actually a small number relative to all of the cancers caused each year. Viruses, including these 2, but also including herpes, human papillomaviruses, hepatitis B virus, together cause about 15% of cancers worldwide. Importantly, EBV and KSHV both persist in cells as plasmids. That means their genomes, their DNAs, are physically independent from cellular DNAs. And that's going to be important throughout this talk. So let me just point out that other viruses, HIV, for example, makes a DNA copy of itself, and that DNA is maintained within the cellular DNA. EBV and KSHV have their own DNA elements, and they keep them separate from the cellular DNA. Both of these viruses, when they infect cells, can promote survival of the cells and or proliferation of those infected cells. And the significance of this statement is that if you think about it, those are really properties shared by tumors. Many of you will know of EBV because it causes infectious mononucleosis. So let me describe briefly what that disease is. It's a disease in which B cells, you're going to hear me talk about B cells, those are the cells in us that make antibody molecules. It's a disease in which EBV infects those B cells and induces them to proliferate. Almost always, there's a very powerful immune response that recognizes those infected proliferating cells and kills them. So it's the summation of infected B cells growing and the immune response killing them off that is infectious mononucleosis. I'm going to focus on describing our work on EBV first. It was discovered in the 1960s, and then I'll turn to Kaposi sarcoma herpes virus. It was discovered about 30 years later. So what does EBV look like? Here you see an electron micrograph of individual Epstein-Barr virus particles. And the dense core actually represents, in this case, the DNA of the virus. It's surrounded by a protein layer. And then that protein layer is actually surrounded by a lipid, which is really fat. So I want to put this virus particle into a context and put the DNA into a context too. Actually, their sizes. So if you were to take, say, 500 or 1,000 of these complete particles and line them up in a row, the length of that row would be about the diameter of one of your hairs. So these are way too small to see with your eyes. They're actually way too small to see with a conventional microscope. But they have a lot of power. Let me put the DNA into context for you too. If you were to take out the DNA from the core of one of these particles and stretch it out, it would be about 3 to 5 times the length of a nucleus of one of your cells. Now, you may not all immediately have a feeling for how big a nucleus is, but you're going to see a lot of them later on in these slides, and I'll show you. But if you were to take the DNA out of that nucleus in which EBV resides, the cellular DNA, and stretch it out, it would be about 2 to 3 yards in length. It dwarfs the length of the EBV DNA. It's about 20,000 times longer than EBV DNA. You know, so at some level, these are just gee whiz numbers. But they're more than that, because these numbers reflect the information encoded either by the virus or by the cell. And the cell's got 20,000 times, roughly speaking, more information encoded in its DNA than does EBV in its DNA. The significance of this is that EBV with this tiny amount of information can dominate the cell dramatically. And in effect, what much of our research and the research of a lot of other people is focused on is trying to understand how this small amount of information can so commandeer or dominate the infected cell. So what does EBV do to a cell? How can we study it? So first, let me tell you how we get the cells to study. If we take blood from you, and let's start with about 10 cc's, and that's 2 teaspoonsful, we can purify from my blood or your blood B cells. Again, those B cells or B lymphocytes are the cells that make antibody molecules. And if we get 10 cc's of my blood, I'm not very good as a donor, but if we do, we can get about 100,000, maybe, say 100,000 B cells from that 10 cc's, those 2 teaspoons of my blood. Put them, actually do it from 100 mils, take 1,000,000 cells and put them into this petri dish in this diagram. Feed all of the normal media you need to keep most cells dividing and be happy. And look what happens to those B cells. What happens is nothing. They sit there, and then over time, they just die. So these cells are not capable of proliferating under the means that we grow most cells in cell culture. But on the right of the slide, you'll see what happens when we add EBV to them. You can infect greater than 90% of these B cells with the virus. And then what happens, and that cartoon is really showing you when we've taken infected cells and then we've embedded them in a gel like Jello. And now you can see individual cells growing out as clumps. And in a regular microscopic image shown below, you're seeing the proliferating cells. They are proliferating because they were infected with EBV. And this, in part, is the biological phenomenon we'd like to understand. So let me ask, how might you understand it? For example, you can imagine that the virus gets into these cells and it flips a switch. And once that switch is flipped, these cells are destined to proliferate, to divide, to give rise to progeny. Or you could imagine that the virus encodes information that's necessary to maintain them in a dividing state. And then this plasmid DNA, this DNA that is distinct from the cellular DNA, somehow has to get passed on to the progeny cells. So this is what we wanted to understand. And I'll describe a first set of experiments to understand it. And they were conducted by Asuka and by Arthur. So Asuka was a postdoc in the lab, and now she's a professor in Nagasaki University in Japan. And Arthur was actually an undergraduate at Wesleyan, and he was in astronomy. So he learned a lot of computational work there. And in particular, he learned how to process images from telescopes on photographic prints. It turns out that's not very different from what you have to do to understand images in microscopes. So he helped us that way. So Asuka wanted to understand if she could actually visualize EBV DNA in a live cell. And then if she could ask the question, does it get distributed to daughter cells? Is it synthesized? So how might you actually see DNA in a live cell? I've shown you it's way too small to see with a regular microscope. It's way too small to see with a fluorescence microscope. So what Asuka did is to take EBV DNA and engineer it so that it then had little pieces of DNA from bacteria, a specific DNA. And that DNA encodes, or that DNA will bind a protein from bacteria. That DNA is not in normal mammalian cells or in human cells. That protein is not in normal human cells either. She could then take that protein and engineer it to be a fusion with a fluorescent protein, green fluorescent protein. So she could take EBV DNA now that could bind that protein. That protein was fluorescent, and she could see EBV DNA in live cells. So let me show you some of her images. And they're quite beautiful, I think. And now you see a nucleus. So I promised you to see one, and this nucleus is probably about, the scale bar is 10 microns, so maybe it's about 20 microns across. What you can also see are these two dots. And those dots really are reflecting EBV DNA. Because Asuka put in 256 binding sites for this bacterial protein called the LAC repressor, because there's such a high local concentration of these binding sites, there's a high local concentration of GFP, and in the fluorescence microscope, you can see these two dots. They represent EBV DNA. Now, watch what's happening. It's quite lovely. She took these images just after cellular DNA and viral DNA had been synthesized, just before cells divide. So they divide in mitosis. They do so in an orderly way. And so at the end of these 5 slides, you can see this 2 daughter nuclei. 1 here, 2 there. Now watch what's happening to these signals from EBV. They're 2 just before the cell begins to divide. They're 2 just a little bit later, but now look at them. They're really 4. So these signals back here are actually pairs. And then these pairs are dividing in the middle of mitosis during the time in which cells divide. And you can see them now associated with 2 daughter nuclei, and here in 2 daughter cells. So from this, we can say EBV DNA, all of that DNA was already synthesized by this time in the cells, but that it was co-localized as pairs those pairs divided during mitosis. They separated as chromosomes separated to daughter cells, and they were distributed evenly to the daughter cells. So this was a lovely find. Oskar built on it, and I'm going to show you another set of her images when she followed this same process in 31 cells. She followed it if I can advance my slides. I can. She followed it from now the entire cycle of cell division. So at this point on the left, the cells have already divided. So you're looking at one nucleus with one signal. And then at this point, the cells have divided again, and you're looking at two daughter cells. She measured the intensity of the signals, and that's shown in these cartoons below. So in this one example of 26 cells that she followed, to make it easier, she chose cells that had only one signal. That one signal eventually becomes more intense, about twice as intense when it's duplicated. Then when the cells divide and distribute these plasmid DNAs, you see each daughter cell gets one, and again, the intensity is dropped down to about half. So in these 26 of the 31 cells she followed, EBV DNA was duplicated at the time when cellular DNA is duplicated, and it was distributed faithfully to daughter cells, just as one might expect. But look over here. 5 of these did something that was completely unexpected, that was exceptional. Again, the cells already divided, and you can see a single signal of this particular intensity. But in S phase, that signal does not get duplicated. That intensity does not increase. In fact, there's no synthesis of EBV DNA. And when this one cell divides to give rise to 2, only one daughter can get that plasmid. So in 5 out of 31 examples, EBV DNA was not being synthesized. So what does that mean? It means that about 16% of EBV plasmid DNAs fail to be synthesized each cell cycle. And that really leads to 8% of the daughter cells having fewer replicons, fewer plasmids than their parents. Now, I just want to dwell on this for a moment. And let me point out that Arthur used these numbers and simulated them in computer simulations, predicted what would happen to other cell types than the one that Oskar was studying. And those simulations predicted what then Oskar and Arthur found. So all of the cells that have EBV plasmids will lose some of those plasmids each cell cycle, each cell division. And what that means is, if you think about it, cells, doesn't matter how many plasmids are there initially, if there's destined to be an 8% loss per cell division, then over time, these proliferating cells should have no EBV DNA in them. And that leads to the implication that EBV will be lost from any population of proliferating cells unless it affords them a growth or survival advantage, so that those cells that maintain the viral plasmids outgrow those that lose them. So that implication is really important. How do we test it? So you can imagine probably multiple ways to do so. I'm going to describe work done by Dave Ferrati, who was a graduate student. When he graduated, he became a postdoc here in Jamie Thompson's lab and really worked on stem cells, in particular stem cells that give rise to the arterial cells that make up our blood vessels, and now has moved to a company, Emoji Biofarm in Colorado. So Dave wanted to test that implication, because if the implication is true, then we really know that you've got to maintain EBV DNA in the cells for them to grow as tumors, for example. So how might he do that? How can he test whether you need to maintain EBV DNA in cells for those cells, for example, to survive and proliferate? So imagine the following. This just depicts a simple cell. The circular dots in here represent EBV DNA. What Dave wanted to do is to find some way to force the loss of those plasmids, those EBV plasmids from these B cells. And in particular, he worked with Burkitt lymphomas, the tumor that was first associated with EBV back in the 60s by Dennis Burkitt, actually back in the 50s. So how might you force the loss of EBV DNA? Well, we and other folks had learned that this EBV DNA is tethered to cellular DNA. And that's in part the way it gets passed on in mitosis to daughter cells. You can think of this as EBV DNA as piggybacking on chromosomes. And then when the chromosomes get distributed to daughter cells, it goes along for the ride. The way it's tethered to the cellular DNA is through a viral protein called EBNA1. So EBNA1 binds to viral DNA and it binds to cellular DNA and it brings them together. What Dave did is to take this viral protein and engineer it so although it could bind DNA, it could actually bind EBV DNA, it could no longer bind cellular DNA. And it would compete for the normal EBNA1 protein binding to both cellular and viral DNA. So if you made a lot of it in cells, it would win. So he then made Burkitt lymphoma tumor cells that would express this derivative that would inhibit EBNA1 from tethering. It would block tethering. And then you could ask the question, what happens? Because that would lead to a loss of EBV DNA over time. And this is what he found. So he's doing this with Burkitt lymphoma cell lines, but he did it also with cells that arose from people who had tumors arising after immunosuppression and transplants. And in all cases, he found the same. So what you're looking at here on the x-axis is days. And what you're looking at on the y-axis is doublings. How many cells did he count? He counted them by hand. And this is actually an exponential. He goes 0, 5, 10, 15, 20, 25. So if you had one cell down here, after 10 doublings, you'd have 1,000. After 20 doublings, you'd have 1,000,000. And the black line is representing the growth of those cells without the inhibitor turned on. And what you can see is they're growing very, very well. So that take over here, by about 22 days, each cell that was down here now has given rise to 1,000,000 progeny. So they grow well. Look what happens when he turns on expression of the inhibitor. That's the inhibitor that's going to block tethering. EBV DNA is going to be lost from these cells over time, and he proved that it was being lost. I won't show that data, but he proved it. That's shown in this red dotted line. And what you can observe early on is there are fewer cells, and then there are fewer cells, and then there are fewer cells, and pretty soon there are almost no cells, and they're dying. So what that tells us is that you need to have EBV DNA retained in this tumor cell for this cell to survive and for this cell to proliferate. And we therefore know that if cells maintain EBV DNA, as do all the tumors with which it's associated, then EBV is providing them some real important advantages, growth and survival. So we wanted to ask, is this also true for Kaposi sarcoma herpes virus, KSHV? And set out to do somewhat parallel experiments. And they were done by Yafang and Catherine. So what they wanted, so Yafang was a postdoc in the lab, and now she is an assistant professor at Chongqing University in Taipei, Taiwan. Catherine was a graduate student. After she graduated, she worked at Prairie Microscopy, where she became expert in all forms of microscopy, and then came back to UW and now is providing us her expertise in the fluorescence activated cell sorting core. What they together wanted to do was to ask, could they visualize KSHV DNA really as Oscar and Arth had visualized EBV DNA? So use a similar approach. And they were able to make this approach work. So again, they took KSHV DNA, they engineered it to have multiple binding sites for this bacterial protein, the LAC repressor, then expressed in these cells, the LAC repressor fused to a color. This would be, in this case, probably RFP, red. And then they could look at cells over time, live cells, to follow what happened to those signals. And I told you that KSHV looks in an electron microscope just like EBV. It does. They're similar viruses, they're herpes viruses, and you would have expected this is all going to be the same, and it wasn't. It's a lovely finding too. So let me describe what they saw. So here, by this time, by the way, Arth had worked out how to measure the intensities of these signals quite accurately. So what you're looking up here are the signal intensities arbitrarily as thousands. And that's in a cartoon that reflects the cells that they're following over time as shown below. And there are two sets of them. So actually this B1 gives rise to B2. But we can just focus, for example, on this bottom layer to make the point that I'd like to. So you can see the signals. And what you can see is when cells divide, they don't necessarily get a signal. The signals are duplicated in S phase. And again, at cell division, some of those cells may not get any signals. You can also see from the intensities of these signals, let's say this is roughly 100,000, that's 10 times what you can see in some signals up here. And since the signal reflects the intensity of individual DNA molecules of KSHV, you can say that this signal's got about 10 times as many plasmids as does this signal. So really, Yafeng and Catherine learned two dramatically new observations. One is that opposed to EBV, KSHV distributes its daughter molecules to daughter cells as clusters. So these are big clusters that are getting distributed. And it does so not faithfully at all. So that's really two ways that these proliferating cells can lose viral DNA. So again, Yafeng and Catherine found that KSHV plasmids are lost from cells both by failures in DNA synthesis and by the random partitioning that arises from viral DNAs forming clusters. Completely unexpected. The implication of their finding, again, is KSHV plasmids will be retained in a population of proliferating cells only if they afford cells a growth or survival advantage, so that cells that inherit these viral plasmids outgrow the sister cells that lose them. As with EBV-associated cancers, KSHV-associated cancers, when you look at one tumor, effectively all of the cells have KSHV in it. That's telling us that the tumors that are associated with KSHV are really caused by it. So KSHV was actually first identified in about 1992, 1993, something like that. And very shortly after it, the tumor that I'm going to describe for you now, primary effusion lymphomas, was detected and was found to have EBV DNA in it. And so people really thought, okay, it's caused by KSHV. But it also had EBV DNA in it. And you can imagine from all that I've told you so far, we're quite convinced that if any tumor has both EBV and KSHV DNAs as plasmids in it, both of those viruses are contributing causally to this tumor. So we wanted to study it in detail. And this was carried out by Aurelia and Mitch. So Aurelia was a graduate student. She came from France. She's, after graduating, returned to France. She now lives in Paris and works with AstraZeneca on SARS-CoV-2. And Mitch and I have worked together for more than 13 years. And Mitch can do anything anybody else in the lab can do. But also, he's really mastered now bioinformatics. And what bioinformatics is the application of really top-notch computer skills to understanding biological sets of information where there's a huge amount of that information and can only be studied by computationally. So what they wanted to do is to develop a model to study primary effusion lymphoma and study it both from its inception through its progression. So this is a human tumor. These viruses really only infect human cells. We have to make such models in cell culture. Let me show you how and what they did. So first, primary effusion lymphomas, I'm going to call them PELs. They are a monoclonal expansion of B cells, meaning it's the progeny of one cell. They constitute about 3% of AIDS-associated lymphomas. They're rare. And they're usually co-infected with both viruses, KSHV and EBV. So what Aurelia set out to do is to ask the question, could she model PELs in vitro? And I show in the diagram what we mean by that modeling. Could she take these peripheral B cells that you can get, remember, from about 100,000 of them from 2 teaspoons of my blood, and now infect them with both viruses, such that these viruses begin to grow and they're maintained in their growth. These red Ys represent antibody molecules. Remember, these are B cells. They secrete or they synthesize and secrete antibody molecules. And in this diagram, I have KSHV shown in green and EBV shown in white. Why? Well, because Aurelia used a really valuable tool. She used KSHV particles that were engineered to express the green fluorescent protein, the very protein that Oskar and Arthur were using to study when fused to the LAC repressor, EBV DNA being distributed to daughter cells. But in this case, the KSHV just encodes and expresses GFP. So if a cell is infected by it, it's green. You can see it's green by fluorescence activated cell sorting or in a fluorescence microscope. So could she reproduce co-infection, co-transformation, such that these cells would grow out? And then they could be studied as a model for PELs. But please remember, I've already told you that EBV can infect this cell type and can infect about 90+% of it, and it induces proliferation, as you've seen, of these cells. So these experiments were going to be made quite difficult by the fact that EBV can also do what we're asking each of these viruses to do. Could Aurelia make this work? And the answer is she could, and it was quite lovely. And her results are shown here, and I want to walk through them slowly because the numbers in them are so important. So again, she's taking primary peripheral B cells and exposing them either to KSHV or to EBV or to both. And I've made the assertion up here that EBV infection supports efficient infection of peripheral B cells with KSHV. And you folks can decide whether you think it's efficient, but let me go over the numbers. So first, Aurelia found that if she took these B cells and exposed them only to KSHV, she could get no infection in 5 out of 5 trials. If she exposed them to EBV, and then 3 days later exposed them to KSHV, she could get no infection with KSHV, as shown by no green cells. Only if she infected or treated these cells, exposed them to both viruses within about a 24-hour window, could she get those cells to be co-infected. And the fraction or percentage of the cells she got infected were on the order of 1 to 2%. So a very small quantity or fraction rather of these cells could be infected. Recall, if I can get, say, 100,000 of my B cells from 10 cc's, 2 teaspoons of my blood, and you can infect only 1% of them, you're down to 1,000 cells. And following 1,000 cells may sound like a lot, but it's not. That's not an easy thing to do. And more importantly, when you've got 1,000 of them co-infected, you've got another 900,000 infected with EBV alone. And so they're going to outgrow everything. And they did. So making these experiments work was not only a tour de force, it really required being able to select out those cells that were green, those cells that were infected with KSHV and EBV. Because we learned that every cell that could grow that had KSHV in it also had EBV in it. So it's a beginning of a model to study primary effusion lymphomas in cell culture. What can we do to expand on this model? And this is really work that Mitch has done and then carried it out with Wei Wang and Paul Lambert here in McArdle. So let me describe these results because they're quite marvelous. What Mitch and Wei did was to take the co-infected cells, cells that had both EBV and KSHV in them, along with a fraction of cells that had only EBV in them, so mixed populations, and then asked, could they grow as PELs grow in people? they grow in the body cavity, in our peritoneal cavity, for example. So Mitch and Wei introduced these cells into the peritoneal cavities of mice, NSG mice that are so immunocompromised that they will accept human cells and allow them to grow. Mitch could monitor which cells had EBV DNA, which cells had KSHV. So over here, EBV is shown in these green dots, KSHV is shown in these red dots, by using probes for these DNAs that were sequence specific and coupled to fluorescent dyes. So he could count what fraction of cells were infected with KSHV, and recall if they've got KSHV in them, they always have EBV, and what fraction of cells had EBV only. He took these mixed populations before they were injected into the mice. So before, something close to 100% of them were EBV positive, and less than 10% were KSHV positive and EBV positive. 28 days later, he could harvest tumors along with Wei from the peritoneal cavity of these mice. And now look what happened. Now, this 8.7% has grown to be 76.1%. That is, these are doubly infected cells, they're growing as tumors, and the doubly infected cells grow much better than do the EBV cells alone. So because, and you can see that no more than 25% of these total could be EBV alone, and before it was close to 90% being EBV alone. So by propagating these cells that were co-transformed in vitro in the peritoneal cavity of NSG mice, we've learned that they grow better than cells infected only with EBV, that KSHV is providing these cells some selective growth advantages in the body cavity, which is really quite characteristic of what happens with primary effusion lymphomas. So we are very keen to pursue studies of this model to help us understand early events in the formation of primary effusion lymphomas. So let me summarize. EBV and KSHV plasmid DNAs are maintained in proliferating cells only if they provide those cells selective advantages. Both viral DNAs are in most primary effusion lymphomas. Both must be causing this lymphoma. And by that I mean, if you look at the tumor cells, those tumor cells have each of these DNAs. Each of these viruses is making a contribution to those tumors. They cause them. Both together allow some infected B cells to grow in vitro and to grow preferentially as tumors in the peritoneal cavities of mice. And this we want to study further. Thank you.
Wow, that is really fascinating. And not only did you present the information in a easy to digest format, I just want to give a nod to you, how thoughtful you are in giving a nod to your colleagues and your students in the, and I know you're going to say they do all the work, right? But
no, we do it together.
Yeah.
It's amazing.
Yeah, but that's just
it's
so
a pleasure.
thoughtful, and it's very clear how much you enjoy teaching. You do a very excellent job of presenting. So thank you. I don't want to put you on the spot, but notable. Hello, everybody. Fran Palaio-Moyer, Badger Talks producer, and we're talking with Dr. Bill Sugden about the implications viruses have on certain cancers. Feel free to post any questions in the chat. I'm going to kick off with a couple of questions. So if I'm just an average person, not of a medical background, watching this, what does this mean for us in our doctor's offices? And potentially, do we see something resulting from this when we go for our exams if we maybe had mononucleosis at one time? Or how do we even know if we have the KSHV? Like, will there be testing, do you think? Or how do you see this manifesting in the office?
First, I think that for the vast majority of us, there are effectively no consequences of what I've described to you. But for those of us who are immunosuppressed or immunocompromised, then we should worry. So, and there are times when that happens for us all. If you're going to have some sort of kidney transplant, you're going to be immunosuppressed. It's at times like that that people have to worry about infections with these viruses causing cancer. I mean, I'm making broad stroke generalizations, but really for both EBV and KSHV, unless if people if they're infected with HIV, you got to worry. But really, in this society, these viruses are primarily going to contribute to cancers in people who are immunocompromised. That's not going to be true in other places in the world, but it's true here.
Okay. And maybe I just need clarification on this. As we age, does that virus naturally diminish in our cells, or?
No, definitely not. So two viruses, Epstein-Barr virus and another member of the herpes virus family, cytomegalovirus, are with us lifelong. So you almost certainly are infected with it. I know I'm infected with it. And you can ask the question, how does our immune response when I'm 77 years old, in me, what does it do? Turns out a lot of my immune response is devoted to limiting the infection or the consequences of infection by these two viruses, Epstein-Barr virus and human cytomegalovirus. And in fact, that's one of the reasons probably that people at my age are less competent to respond to other kinds of infections, because we've devoted so much effort to limiting these two herpes viruses.
See, that's very interesting. Thank you. You had mentioned at one point in the presentation about research with other labs, and we did ask the question of Dr. Deming last week as well. Can you talk a little bit about collaboration on research with other labs?
Sure. So I mentioned work by Wei Wong and Paul Lambert that we're doing with them, and we are working with them. So collaboration has both a plus and a minus. The plus is you can do so much more. You have real expertise from people doing things they can do that I can't do. On the other hand, a little bit, it's a cost in that graduate students ideally will have their own projects, and they're the bosses of those projects. So their collaboration has to be done in such a way that you're not impinging on their education. But otherwise, it's incredibly valuable.
Thank you. We often hear that teachers will grab these Badger Talks Live and share with classrooms. Could you tell us a little bit about your, in a brief mention, your professional career growth, and how did you end up in cancer research, and also why UW specifically?
Okay, I can answer both of those. So as a child, if I read anything in the obituaries and somebody had died of cancer, it would be described as they died after a long illness. Cancer was a word that would not be mentioned in the newspapers or in general in public. And pardon me, that's crazy, because we should know about it. We should know how to limit it. We should know how to prevent it. We should know how to treat it. And so growing up in that era led me to want to do something that was cancer research. And I began working in what's called chemical carcinogenesis in college. And then through a very circuitous route, I eventually started working on viral-induced cancers. Why did I come to Madison? That was a long time ago. It was actually in 1974 that I looked for jobs. And I looked at big name universities on the East Coast, Harvard, MIT, Princeton. I got job offers there. This was the only place where faculty really cared about cancer research in McArdle. I wanted to come here, and I was lucky enough to be able to do so. And here I still am.
That's a beautiful story, and we're really glad you're still here doing this important work and leading so many others in the effort. Thank you so much for joining us today.
Of course. Thank you.
I also want to give a shout out to Emeritus Professor of Oncology, who I understand had a birthday yesterday, Dick Burgess. He was an important part of getting this series together. So I just want to say thank you so much to him for that. Please also tune in next Tuesday, September 27th at noon. We're going to be continuing the focus on all of this amazing research coming out of the McArdle Lab. And we're going to be talking to Dr. Wei Zhu, who's another professor of oncology. And she's going to be talking about the latest research as it relates to breast cancer. So please tune in, share with your friends. we'll look forward to seeing you next week. Also, check out the Badger Talks Live website at badgertalks.wis.edu. You can see the upcoming schedule of talks coming up. Sign up for our email list and be the first to know about the talks. Also, search the roster of over 400 UW faculty and staff who've signed up to give talks around the state in communities like yours. Thanks for tuning in, and we'll look forward to seeing you next week.